So I thank you for staying for the next succession. I guess it's part 2. So I'll build up on some of the stuff Simon was talking about. I'm more of a molecular biologist, so I really like to study what the molecules do, in particular what happens to the DNA during recombination. I noticed that my title is not very flashy, so I kind of put in a little bit of more information about DNA repair, trying to lure or hoping that you stay for more on this. So Simon talked about these double-strand breaks that are necessary to start the recombination process in meiosis. We all know that double-strand breaks are also introduced randomly by outside agents like ionizing radiation, free radicals, also some intrinsical elements in our DNA like transposable elements or long repeats might introduce double-strand breaks. And double-strand breaks are really bad for the cell. They induce apoptosis, aging. Even if you have gone to some of the cancer talks, they all are associated with double-strand breaks that are going berserk and usually then have a cancer phenotype. They also produce cell arrest for good reasons. But there's 2 biological processes that use these double-strand breaks. One you heard about, meiosis, the other one is the immune system. So how does a cell, or why does a cell now use these double-strand breaks that are really bad for a very important biological process? So this actually adds some of the complexity of meiosis. And I have to say that these double-strand breaks are really programmed and very tightly controlled. We heard a little bit about the control with PRDM9. This shows also the stage of when and how the double-strand breaks are introduced. So we are in prophase 1 in the first stage of meiosis, where the DNA is not soup of spaghetti, as I like to tell my students, but more already complex in a more high structure order where you start seeing these loops and an axis that will then mature in the synaptonemal complex. And you have this double-strand break machinery associated to the axis, and the breaks themselves happen in the loop. So there's a very complex process that brings in the loop into the axis, and we call this loop axis complexes or loop axis tethering that was already described in the 90s by Nancy Kleckner. And it's only this loop tethered to the axis where you have this double-strand break. Simon already talked a little bit about the importance of SPO11 itself, where you have a series of different proteins actually introduced part of this recombination initiation machinery, but actually it's the SPO11 endonuclease associated with also topoisomerases that then cleave the DNA, and now we go to a very simple form of the process itself, where we have resection. You have also the different proteins then binding to these resected 3' ends that are now very reactive, I call it, and they start being, they want to be repaired immediately. When you have double-strand breaks in somatic cells, these can be repaired also, as we know, with the CRISPR-Cas system, either by non-homologous end joining, but if you have a homolog nearby, then you go and repair it by homologous recombination. And this is exactly what meiosis tries to do. It tries to repair these double-strand breaks by preferentially homologous recombination, and this produces, and I skipped a series of steps here, but mainly you can produce crossovers or non-crossovers, as also was nicely presented in the previous talk. And you end up with, in sperm at least, 4 different recombination products, one with a crossover, or also sometimes just with a non-crossover. And again, Simon presented this way better in terms of within a crossover you have also a series of small non-crossover regions. Now, you have to think about this also, that in the males, all 4 meiotic products are being or can be used as gametes. In the females, it's only one of these four products that actually becomes the oocyte. So there's also this big difference. So in order to understand a little bit recombination, there has been several methods of how we have learned of where the hotspots have been. So the question of where has been actually taking us many, many years, and this has been the work of the past 100 years, I guess, starting with Mendel, trying to see, and with Morgan, trying to see where recombination happens. And Morgan with Drosophila, he used pedigree analysis, and now we still do pedigree analysis to see where these recombination or where these crossovers might occur. And it's very simple. You have mom, dad, and then you have a series of offspring. Maybe you have a second and a third generation. And then by phasing the two haplotypes, you can see whether there has been a switch in phase, and that's where your recombination will be. Now, I'm bringing this up because these different methods give us a different picture of recombination itself. So first, there's a resolution issue. So as you can imagine, here we just only got one crossover, and the location of this crossover so is within megabases of information. As you keep adding on the pedigree, you can add more crossovers to this and locate to a finer scale where the crossovers or the non-crossovers are occurring. And as we keep having more data on human pedigrees, doing whole genome sequencing, or also doing whole genome sequencing on crosses of mice, as Simon has been doing, we start getting more information at a fine resolution of where these crossovers happen. I think the best analysis, now published in 2019, gets us a resolution of 600 base pairs. It has the advantage that it represents active recombination, so it's actually happening now, and it's taking into account both male and female events. Then we have what we call double-strand break mapping. It's basically taking the proteins involved in the recombination initiation machinery, either SPO11 or DMC1, co-immunoprecipitate those proteins, and then sequence the DNA that is attached to those proteins. This is done using testes because you need a lot of DNA material, and that's why we call it's representative of active male events. And SPO11, at least SPO11 sequencing, gives us an incredibly high resolution because these small oligos are about 50 to 100 nucleotides long. You can even see where the upstream and downstream oligos are, so you have also a directionality. But unfortunately, we only have a SPO11 map for yeast and for mice, not for humans. Then we have, although we do have a DMC double-strand break maps for humans. And then the last method coming up once we sequence the human genome, and then we genotyped it with HapMap. The group of Peter Donnelly, together with Simon Myers, actually started to categorize those genotype information from HapMap into haplotypes and trying to link those haplotypes, trying to extract the information of where haplotypes are broken up. And they found that with a very complex mathematical analysis called coalescent analysis, that some regions are broken up more often than others. And these were called then recombination hotspots. Now, because genotype information represents the information over many thousands of generations and represents an average, it represents actually a population average, and it's what we call historical recombination. So as you have these LD maps or linkage disequilibrium maps, they have a very high resolution, in fact, but they do not represent active recombinations, but events that happened long time ago. And there's a fourth method that we have been developing. Actually, Alec Jeffrey's group also have been using this method, and it's called pooled sperm typing. So the idea is that you take your sperm, which are meiotic products, and you pool them such that you have on average one recombinant or less on a small pool of DNA. Then you use special PCR tricks in order to amplify only the recombinant DNA molecule. And you have to do this in several rounds such that you have a big difference between a single molecule that has a recombination event versus all the other pools that are empty. So basically the amplicons that we get represent copies of a single recombination event. And then you can sequence this or genotype this and really understand what's happening here. Now, unfortunately, this is a very laborious process, so you can't just look at a single hotspot or a couple of hotspots at a time. It's a lot of, still a lot of handwork, and so we call it active recombination. It's only male recombination, but it currently is the method that gives you the highest resolution in terms of being able to distinguish events that are maybe only 10 to 20 nucleotides apart if you have the informative SNPs. So also the nice thing about pooled sperm typing is that you can both assay crossovers, and again, if you do enough reactions, you then can count where your break point was. So basically you have a bunch of informative polymorphisms that tell you whether you're mom or dad, and you can then use that information to see where the crossover breakpoints are and fit a Gaussian and say, okay, this is the recombination center or the recombination hotspot. You can also do this for non-crossover, so this is a great assay to then see how the molecules for conversions or non-crossovers work. So most of our work has been based on these types of assays, just trying to understand how the DNA changes with recombination. So one of the first questions I tried to answer is how is recombination organized in the genome? Not only me, but the field. And as already Simon mentioned, recombination is localized in hotspots. When I started this work in recombination a long time ago, I took a random segment of the genome and measured it with sperm typing, and indeed I found some intervals having a high recombination event. And then I overlapped the just published data of LD-based data and noticed that those regions where I measured high recombination with sperm also overlapped with the regions with high LD, with exceptions, and this is where I think these are maybe regions where recombination has been lost, or these are recombination regions that are population-specific. We also learned that the human genome is full of these recombination hotspots. You have seen this from Simon's talk beforehand. And right now we count about 34,000 hotspots and 39,000 double-strand break regions in the human genome. So the nice thing about these studies is that we have maps of where recombination is localized in the human genome. So if you're wondering whether you have a particular region that has a recombination hotspot, you can actually look up those coordinates. What are the properties of these hotspots? Well, they're usually very small intervals, about 0.5 to 2 kb long, separated by long flanking regions that are recombinationally cold. And the thumb of rule is that you have about a 10 or more recombination frequency in these regions. I like to call the motif that is being recognized by the recombination machinery, specifically PRTM9, as a Myers motif, since Simon actually was the first one to describe it, at least in humans. And what we also know now about recombination hotspots is that the females have more recombination, they have more crossovers, they have a longer genetic map. And we also start seeing effects where females have less recombination at subtelomeric regions versus the males have a much higher increased recombination exactly in those regions. So there's very specific differences between males and females, not just only hybrid sterility, but also in terms of when and how recombination happens. And, right. So, what are the factors that determine these recombination hotspots? So you have heard about this trans factor PRDM9 beforehand, and this actually drives recombination in mice and humans, and we believe in a series of other mammals, but there's also a series of other species which don't have PRDM9, and we don't know what drives recombination hotspots or what places these hotspots in the species. And there's a series of other factors, for example, specific motifs or maybe increased GC content, promoter regions, also that we still don't really understand, but we believe that this is kind of an archaic recombination machinery, and PRDM9 kind of is working together with this machinery or on top of it in order to change where these hotspots are located. Now, interestingly, so I showed you this one chromosome map with thousands of hotspots. This doesn't mean that each cell gets as many double-strand breaks. In fact, the double-strand break number is really constrained, the double-strand break number per cell. So each cell doesn't get more than about 200 breaks, and of those, only 10% result in a crossover. All the rest of those breaks are being repaired as non-crossovers. Also, what's interesting in this table is that species without PRDM9 that have and that also have hotspots, but have a much lower number of these hotspots in the genome. Yet the number of crossovers is still about the same. And then you have things that have no hotspots, like Drosophila, which still have a number of crossovers, although reduced. And you have things like yeast that have much, much smaller genome sizes, about the same number of chromosome numbers, and also same number of double-strand hotspots, but a much fewer number of or about the same number of double-strand breaks than very or much larger genomes like they're found in humans or in mice. So the next question that we have been really interested in is, how does recombination change the DNA sequence? And particularly, we have been looking at mutagenesis, because for the longest time, the observation has been made that within hotspots, we see that if we compare the sequences between different individuals or between different species at hotspot regions, that those sequences have more changes, or as we call it in evolution biology, they're more divergent than sequences in cold regions or cold spots. So again, this has been an observation made a long time ago already by sequence comparisons, even when looking at the PRDM9 sites themselves. Simon saw that there's also a lot of changes accumulating at the PRDM9 binding sites, and also regions flanking the PRDM9 binding sites are highly polymorphic. So there seems to be an important mechanism changing the DNA sequences at hotspots, and we wanted to figure out what those mechanisms are. So the first thing we looked at, or the first suggestion looking at mitosis or double-strand break repair in mitosis, was that we have translation error-prone translation polymerases associated with the repair of these double-strand breaks. in somatic cells. It was also shown that in meiosis that this repair of the double-strand breaks, remember you have this break, and during this break repair you have DNA synthesis. And also in meiosis, the polymerases in charge of this DNA synthesis are also error-prone polymerases. So one of my first graduate students took on the ordeal to measure the number of mutations in recombination products, and at that, before she started, the plan was that she would find one mutation in 30 crossovers. So she's like, well, that's an easy task. I guess she didn't know that this was not going to be true, so she ended up sequencing almost 6,000 crossovers, which was a lot of work in order to just find 17 mutations. So not a very impressive database, but the fact that we found those 17 mutations already allowed us to understand a little bit about the mechanisms driving mutations in recombination. First of all, we had, for the first time, experimental evidence that indeed we had a high mutation frequency associated with crossovers. In fact, the mutation frequency that we measured, 9.3 times to the 10 to the minus 7. It's about 70-fold higher than the genome average. So we then ask, okay, what kind of mutations are those 14 mutations? And we saw that we had a very strong bias of strong to weak. Strong are the bases that actually form three hydrogen bonds, the guanine and the cytosine, GC, and they all mutate to A or T. And these are usually transitions, meaning that you have G mutating into an A, or if you're in the complementary strand, a cytosine mutating into a T. And we had this very strong bias of strong to weak. Not only that, we looked a little bit closer where these mutations occurred, and they're all, or most of them, 60% of them, 2/3 of them, happened in the context of a CPG site. So now considering the CPG sites and the number of effective sites, we estimate a mutation frequency of about 10 to the minus 5. Now, the DECODE group now decided to do a similar study. Of course, they have very different scales of data, and they didn't look at 6,000 crossovers, but 4.5 million crossovers, and found 139 de novos associated with these crossovers. The nice thing is that they got about the same answer, that the increase in mutation frequency is around 50-fold higher than genome average. And they also now have information that this happens not only in males, but also in females, a little bit less in females, but as you go away from the recombination hotspot, the number of de novo mutations decreases. So it seems to be a really, these de novos accumulate because there was a crossover there. As I mentioned, these de novo mutations happen in the context of the CPGs, and what this new study also showed was these CPGs were only highly mutable in males, meaning that if you compare the mutation frequencies in fathers versus mothers, you could see that there's only an increase from C to T in the fathers, but not in the mothers. So there's some processes that make CPGs highly reactive in males or highly mutable in males, but not in females. And one of these processes might be the methylation state. So meiosis 1 happens in the adult life in the males. So the germline is highly methylated at that point, versus in females, meiosis 1 already takes place in utero, where the methylation state is much, much less, sometimes maybe only 10 to 20%, although we don't know for sure. So there's a big difference in the methylation state of the DNA at that point. So what we think what's happening is that as you have these double-strand breaks and you have single-stranded DNA, you have these CpG sites that can deaminate, and this deamination event, as you might remember from your chemistry courses, when you have a deamination of cytosine, you produce uracil. If you have a deamination of a methyl cytosine, you produce thymine. So thymine is actually naturally occurring base, so the cell would not know that you have a deamination event. If you have a uracil, this might trigger the repair machinery and have it removed. Also, if you have deamination events of cytosines in the context of a duplex DNA, you form a heteroduplex that also triggers the repair machinery. But in a single-stranded context, you would not have this heteroduplex problem. And also, chemically seeing, you have, in a single-stranded DNA context, a much faster spontaneous deamination rate than if you're in a double-stranded context. And this is much faster in 5-methyl-C. So we believe that this methylation state of the DNA between males and females might explain why you have such a high mutagenesis in recombination. Also, very interesting facts about the de novo mutations accumulated in the females that are independent of CpGs might now be explained maybe by these translesion polymerases. And the fact is that as females age, the de novos associated with crossovers also increase. And we know that the mismatch repair machinery also gets weaker with maternal age. So what we think is happening that these mismatches are not being removed as females age, and that's why they accumulate, that you have more de novo mutation accumulation with age in female recombination hotspots. So I hope that I convinced you that recombination is mutagenic, so that would be one take-home message, that these mutations, as we showed, and also the DECODE group showed, are from strong to weak. So this means that with time, hotspots must become AT-rich. But if you look at the data at hotspots, this is actually the opposite trend. Hotspots have a high GC content. So there must be something else happening during recombination that counteracts these mutation events. So this is something that we looked at further, and we now come to our next question that we're trying to ask, and that is, what's the role of repair proteins in the recombination outcome? So this is another view of what's happening downstream of the double-strand break repair. Here you have strand invasion, you have strand synthesis, and during that you can see that you form crossover intermediates or non-crossover intermediates, and there's one small segment of the genome that we call gene conversion, where you can have the repair of one allele versus the other. So what does this mean? So you have a heteroduplex forming in recombination because of this strand invasion, and this heteroduplex needs to be repaired, because if you have polymorphisms within this region, you will have mismatches. And this attracts actually a mismatch repair machinery. And the problem is, what does a mismatch repair machinery do? Does it remove the red or the blue? Mendel's prediction would be that it removes it such that at the end we have an equal distribution of alleles, so that you have a 50-50 segregation of the alleles. But in fact, or what we often see is that there is a preferential repair of one allele over the other. And this is what we call biased gene repair or biased gene conversion. We also know that the mismatch repair machinery usually prefers to remove the Cs or the As because of these deamination problems. So this is a natural system that will leave the guanine or the cytosine in the strand and remove the adenine or thymine at a AG mismatch or CT mismatch. And this leads to what we call biased gene conversion. And there's lots of evidence by indirect sequence comparison about this biased gene conversion. So what we tried to do, actually, this biased gene conversion has been described in all the way from yeast, humans to mice by different people. And what we tried to do is trying to see evidence for biased gene conversion from sperm typing. So what we basically did was to compare based from our large number of crossover breakpoints where these might be. And what we basically did was to compare, okay, if there's no biased gene conversion, then we should see as many use or unconverted for both reciprocals so that we have this same 50-50 segregation of this particular polymorphism here. If there's conversion, then you see more of these type of products for one reciprocal. And we then calculate a log rate ratio to kind of estimate this. And this is basically the crossover distribution or the breakpoint over distribution of one of the crossovers. And then we estimate this rate ratio here in the log scale. So at 0, it means that the segregation is 50-50. If you have greater than 0, it means that you're segregating more the C versus the T at this position. And if it's under 0, it means that you're under-transmitting the T and over-transmitting the C. And so that now we compared it, and you see that when you have under- or over-transmission that is significant, that it's always biased towards this G or the C. That's the case in all these marked cases. We did a more thorough statistical analysis, and we found that of all our crossovers, 1.2 times there's always a G preferred over an A or a C preferred over a T. This means that in crossovers, there's an overtransmission by 52%. Or in other words, you deviate from the 50-50 expected ratio by a very small amount. So the effect is minimal, but it's still strong. So then we quickly ask, okay, so who wins, the mutation force or this biased gene conversion? And there's a nice equation we could use here, and we estimated that at the end, and of course this depends on the strength of the hotspot, but at the end, all the hotspots, if they continue, will end up with a high GC content, and this is the prediction. But we also know that hotspots don't stay at the same place for a long time, but as PRD9 evolves, they get moved around, and maybe this is a reason of why they get moved around. All right, so last, I want to finish up with a really exciting work about repeat and recombinations. Here we focused on short tandem repeats, and this is a work by another great PhD student that I had. So we had an unusual hotspot with 3 poly-A repeats, some of them being very short, some of them being very long. And we know that within this repeat, we have a PRD9 binding motif, and this is the one that is being used. And this is a double straight break zone. So we had a bunch of donors that were either heterozygote for this region or homozygote for this 1998 repeat. So we have a heterology in the red donors of about 10 nucleotides, either 9A or 19A. All the rest was pretty much the same within this hotspot. So we also found that poly-As have a different nucleosome structure. So if we look at the H3K4 methylation, exactly at the sites where you have long 19A repeats, poly-A repeats, you seem to have less H3K4 methylation, and it's likely because there's no histones to begin with. And there's work by Irene that demonstrate that the longer the poly-As, and it starts with 8 A's, that the affinity to the histone actually decreases exponentially. You also can see that other methods screening for single-stranded DNA also find this region to be open in chromatin. So what does this heterology do? So one thing it does, it actually, if you compare the homozygotes that have no heterology, they're 19A, they have this number of crossovers, but the ones that do have the heterology have a reduced number of crossovers and instead have an increased number of non-crossovers. So it seems that these heterozygotes prefer to repair via non-crossovers instead of crossovers at this particular hotspot. We also see a small reduction in recombination activity overall, and we don't know whether this, how to interpret this. The next thing that we observed is a gap in crossovers. And I guess here where it's being shown, you can see a reduction of it. And this gap in crossover distribution has been also linked for other types of repeats. These are long palindromic repeats. So we were actually surprised to see this with a 10 nucleotide heterology. Another thing that we saw is a reduction in GC biased gene conversion. In fact, here you see that heterozygotes individuals compared to homozygotes have a lower ratio or lower overtransmission of these GCs. And we think this has to do with how this is being repaired. That as you have these heterologies, you form a loop that attracts a different repair system that is not mismatch repair, that cuts out this site, and now you have much longer regions to copy. And this has been also an observation that as you have insertion deletions, you have a higher number of polymorphisms. So there seems to be a correlation there. Finally, want to just quickly talk about the transmission of indels, where you see that certain the small heterologies with 6, 7As, always the 7A is overtransmitted. So the longer the insertion is overtransmitted. But when you have the 19A heterology, you actually cut out the big loop and you actually get always the shorter. So you tend towards deletions. So I will skip this for the sake of time. We finally also looked at complex crossovers where you have small conversion events. Our complex crossovers are pretty similar with other studies. Usually they're short, they involve one SNP, and they're a source of GC biased gene conversion. Now we have a very unusual donor that has a very high number of complex crossovers. It has also a very high number of unrepaired heteroduplexes, and then we looked to see what might be different with this donor, and we saw some variants in repair pathways, repair genes like XRCC1 and ERCC1 that are different compared to the homozygotes and heterozygotes that we have been studying. So we do think that that these proteins might be involved in this repair pathway. So I want to conclude that yes, recombination is mutagenic. It produces 50 to 70-fold more de novos at recombination hotspots, that you have repair bias towards GC or towards insertions. When you have large heterologies, like these poly-As or maybe a lot of polymorphisms in a region, the repair might not be GC biased because it attracts different mismatch repair systems. And this large loop repair actually produces crossover gap. It increases non-crossovers over crossovers. You also have a deletion biased repair, meaning you remove those large loops. And you have an increased number of complex events, which I didn't talk about, but this is also what we see. Complex crossovers occur at a low frequency, but variants of some of these base excision repair and nucleotide excision repair could lead to an increase in these complex crossovers, which is also seen in females with age where complex crossovers actually are also increased. And as I mentioned, females have a reduced mismatch repair. And finally, I hope to convince you that yes, indeed, DNA sequences at hotspots are rapidly evolving, and that's why we have a whole different mechanism trying to counteract this. And last but not least, I want to acknowledge my wonderful group. It has a joy to work with you, and also my collaborators, Andrea and Thomas Ebner, and my funding agencies. And you for staying with me.